Okay, so the next session is From Evidence to Action in Science Policy and AMR Solutions. We have, we're starting off with Serlinda from Queen's University Belfast here. Sorry.
A little too high for me. So I'm going to switch around a little bit here and talk about the host response to fungi as opposed to talking actually about the pathogen. So we're going to talk about the main type of work that we're doing, focusing on C-type lectin-like receptors and antifungal immunity. So in my lab we mainly focus on the immune response to invasive fungal infections. So fungal infections kill around 2.5 million people per year. That's the latest estimates. And I've just highlighted the invasive aspergillosis and candida infections, and untreated, this almost certainly ends in mortality. But you can see even with treatment, the mortality rates are still really high for these types of infections. And in fungi, we're always a bit behind bacteria. So the World Health Organization came out with the fungal priority pathogens list a few years ago. And in the critical priority group, there's Aspergillus and Candida albicans. This list was made due to the mortality levels from these infections, the increasing prevalence of antimicrobial resistance for these various pathogens, and the fact that we need more treatments. So in my group, we've been mainly focused on looking at the host response to Candida albicans and more recently Aspergillus fumigatus. We also look a bit at some of the other Candida species in the high priority group. And generally as a field, as I said, the mortality rates are a lot higher than we would obviously like. So we need better diagnostics, patient stratification systems. So essentially a lot of hematology patients would get prophylactic antifungal treatments. And if we can stratify those patients into more high-risk groups and lower risk groups, so that only the patients who need them are getting antifungal treatments. Potentially the development of immunotherapies to go alongside antifungal treatments. And in terms of fungi, we don't have any approved fungal vaccines. So in my lab, our main focus is really trying to understand the immune response better so that down the line we could potentially try and identify things that could be targeted for immunotherapies. We've also done some patient stratification and starting to collaborate with a colleague to try and develop a vaccine. So I'm just going to give you a bit of an overview of what we're doing in those various areas. So firstly, as I said, we're mainly trying to understand the immune response to fungi. So we've been mainly focused on these C-type lectin-like receptors, Dectin-1, Dectin-2, and Minkle. And when we started all of this, there were papers out about what these receptors did individually in response to fungi. We were looking at Candida albicans, but we didn't know how they all worked together. So in my lab, we created various multi-CLR knockout mice and then infected them with Candida albicans. And so this is with a medium dose with Candida. So the blue line is the Dectin-1 knockouts, which was known. They're quite susceptible. The Minkle Dectin-1 knockout that we made was pretty similar to it, so there wasn't any added effect. But when we knocked out Dectin-1 and Dectin-2, or else Minkle along with it, this is Dectin-1, Dectin-2 double, and then a Minkle Dectin-1, Dectin-2 triple knockout, they were really dramatically susceptible to infection with Candida albicans. There's a little bit of susceptibility at this dose of Dectin-2, but then when we increased the dose to high dose Candida albicans, you can see that the Dectin-2 mice were susceptible. But again, when we knocked out Minkle in combination with Dectin-2, there was no added effect. So in terms of in vivo susceptibility, losing Dectin-1 and Dectin-2 together seems to cause a really dramatic effect. So we looked at the fungal burden. We looked at it with everything, but just showing here the Dectin-1, Dectin-2 double, and then the triple knockout. And you can see at a low dose of Candida albicans, after a week, the wild-type mice had basically cleared the infection, but the double and triple had a huge amount of fungal burden in the kidneys. And so much so that we had these mice all co-housed and blinded ourselves when we were doing the experiment. But whenever we do the harvest, we knew exactly, if it was a double or a triple knockout mice, because you could see these little white nodules all over the kidney. That was the fungus and inflammatory cells. It was very obvious in them. So then we wanted to figure out functionally what's happening in the different cells. So we did an assay where we injected biogel beads into the peritoneal cavity of mice, and then the next morning we did a peritoneal lavage to get, it was basically to recruit inflammatory cells, and then with the peritoneal lavage we recovered the cells. We then incubated them with labeled Candida albicans just for 15 minutes, and then checked on flow how much Candida bound to the cells. So looking at the inflammatory monocytes macrophage population, you can see a nice recognition and binding of Candida albicans to the wild-type cells, and this was reduced in the triple knockouts. And when we looked at all the different knockouts, we didn't see a significant effect, but with the individual single knockouts, you see a little bit of a drop. Then when you go to any of the doubles, so the Minkle Dectin-1 or the Minkle Dectin-2, again, there's another drop. But the biggest drop was then if you lose Dectin-1, Dectin-2, or all three receptors together. So in these cells, it seems that all three receptors actually contribute to that recognition of Candida albicans. In the neutrophil population, however, we really didn't see any significant effect. So it seems that other receptors are doing more of the recognition and binding for the neutrophils. We looked at cytokine production, and again, we did a similar assay. It's using those peritoneal inflammatory cells, and we incubated them with the Candida for 3 hours this time, and then looked for intracellular TNF. And again, in the wild type, we see nice TNF production, and that's reduced in the triple knockout. And again, you're kind of seeing that stepwise pattern in terms of the percentage cells producing TNF. You see a big drop with the double and triple knockout, and then the amount of TNF that is produced per cell is also reduced. And again, that kind of happens. Although with the recognition, that seemed to be that all three receptors were involved. With this, it seems to be there's really just Dectin-1 and Dectin-2 were involved in the cytokine production. So we looked more broadly then at a whole range. We did RNA-seq to look how much was affected by the absence of Dectin-1 and Dectin-2. And we saw a lot of cytokines and chemokines were actually reduced. So then we did an analysis just looking for cytokine, chemokine, and growth factor. So you can see in the wild-type cells, when we stimulate with Candida albicans, you get a nice upregulation of cytokines, chemokines, and growth factors. But this is quite considerably reduced in the Dectin-1, Dectin-2 double, and the triple knockouts. And again, we were able to confirm that for a load of different cytokines and chemokines by ELISA. So we looked at that further then with systemically what that actually meant. And so we used CCR2 knockout mice to look at that. So with these, the monocytes are sort of stuck in the bone marrow and they don't get to the site of infection. And so these mice tend to be more susceptible to infection. And that had been shown, but we confirmed that in our hands. Just looking at, you can see the fungal burdens increased in the CCR2 knockout. And the reason for us looking at this was because we wanted to do adaptive transfers with our monocytes. So we purified wild type or triple knockout monocytes. We used the CCR2 knockouts as the recipient mice, and then we adaptively transferred our monocytes along with Candida albicans infection, and after a few days we harvested the organs. And what we found is that when we added the wild type monocytes, we got better clearance of Candida infection than whereas when we added the triple knockouts, we weren't getting that clearance. So then we looked at cell recruitment. So for this, we labeled, fluorescently labeled the monocytes that we were adoptively transferring, and then 24 hours later, we looked in the kidney. And so what we found actually is that the monocytes that we transferred, after 24 hours, the triple knockouts, we had a lot less of those in the kidney than in the mice where we transferred the wild-type monocytes. We don't know whether that, we injected the same number, but we don't know whether as many of those are not getting to the kidney, if they're not surviving or not being retained there. But what we also saw then was that recruitment of neutrophils to the kidneys was actually reduced. We didn't see any reduction in any other type of cell recruitment to the kidneys. And when we did kidney homogenates, again, we looked for various chemokines, and we saw a bit of a reduction in the CXCL2, which we think is what's causing the reduced neutrophil recruitment. So what we think is happening is that on these inflammatory monocytes, they have really high expression of all three of these receptors. When those receptors are absent on these cells, then the recognition and binding of Candida is really reduced. The reduction of cytokines and chemokines is really reduced, and then inflammatory cell recruitment is really reduced. Whereas with the neutrophils, they are expressed, but they seem to be dependent on other receptors for all of these functions. So while the neutrophil functions do not seem to be reduced, because the monocyte function is reduced in these mice, you don't get the neutrophils recruited to the site of infection. And so then you get overgrowth of the fungi. And then eventually, because it's not cleared at the start, you actually get a huge influx of inflammatory cells and get a major immunopathology role. So using these mice, then we wanted to see, in terms of Aspergillus and some other fungi, does the same sort of thing happen here? Because with Aspergillus, we know that Dectin-1 and Dectin-2 are involved to a certain extent. We don't really know anything about the role of Minkle here. And all of those, and these receptors are important, as I've said, for the production of cytokines and chemokines. So what we've done more recently, and what we're working at on the minute, sorry, that's come out quite blurry, we've done a few different RNA-seq sets with different fungi, stimulated on wild-type Dectin-1 knockout, Dectin-1, Dectin-2 double, and then the triple knockout, trying to see what role these different CLRs are playing. And what we find is that there's a cluster of genes that's really nicely dependent on the different CLRs. And we've taken the Aspergillus data. We also have some data with Rhizopus, and what we find is that the CLR clusters, there's this conserved response, and it seems to be like viral pathways, possibly interferon. So when we put all of those cells into the interferon database, we actually find a huge amount of it is actually interferon-regulated genes. And we've confirmed that with our knockouts, that interferon beta is really reduced. So this is what we're actually working on at the minute is, you know, with the interferon regulated genes, there's a lot of things in there that we know are involved in bacteria and viral pathways. And so we're actually taking some of those genes at the moment and looking at what role they actually play in terms of Aspergillus and Rhizopus. And then I kind of wanted to switch from that into what we've done with patient stratification and are doing at the minute. So the reasons for patient stratification is, as I've said, there's quite a widespread use of antifungal drugs at first suspicion of infection. With hematology patients, many of the hospitals just prophylactically give them antifungal drugs, which all of this contributes to the development of resistance. Additionally, with these patients, you know, there may be chemotherapy patients, and sometimes then if they're being given antifungal drugs, that can interfere with their primary treatment, and sometimes the chemotherapy might either have to be changed or stopped. And then there is quite high toxicity with antifungals. And what was more relevant when we did this study is the high cost. It used to be about £1,000 a day per patient for antifungal drugs. I think that has gone down considerably now because a lot of the patents have expired. But for the study that we did, we got samples from AML patients and bone marrow transplant patients on their first admission to hospital. And the reason we got samples from them is that both of these patient cohorts have a high risk of developing invasive aspergillosis. So we did this at the Heath Hospital in Cardiff, and there they don't prophylactically treat patients, hematology patients with antifungal drugs. So there's about a 27% risk of them developing invasive aspergillosis. So for the blood samples, then we took RNA, we sequenced these CMCLRs that I've been chatting about, Dectin-1, Dectin-2, Minkle, and also MCL, looking for SNPs in them. We also looked at expression of the genes, and then we extracted peripheral blood mononuclear cells, or PBMCs, and stimulated them with LPS as a control, or Aspergillus fumigatus, and then looked at cytokine production by ELISA to see again if there was any correlation with development of invasive aspergillosis. So what we found is that, so these are the Dectin-1, Dectin-2 Minkle genes, and here this is invasive aspergillosis, and this is no evidence of fungal disease. We didn't see any correlation with the CLR expression level. We found a couple of SNPs in the CLRs that we were looking at. One was quite common, this point mutation in MCL, but we didn't find, again, any correlation with development of invasive aspergillosis. We did identify a novel mutation in Dectin-2. We just found this in one patient, and it was a deletion of a base pair. It was homozygous deletion, and so what it actually did was caused a frame shift and then resulted in an early stop codon. So quite a bit of the protein was no longer made, and these are quite important residues. So we collaborated with a colleague to try and do some modeling. So here is the full-length Dectin-2, and this is the ligand mannan. And in orange is the bit that's missing in the Dectin-2 mutant. So there's a beta strand missing. There's some important residues that are important for binding missing. And sort of this goes down the center of the protein. So the theory was that that would lead to an unstable protein. And sure enough, when we made constructs and expressed these, RNA levels were the same between the wild type and the mutant, but the protein levels were much less. So the protein level is in gray here, and the wild type Dectin-2 is in black. So basically, this mutation that we identified led to an unstable protein and didn't bind. So, well, it actually was no longer expressed on the surface of the cell. It was also sort of expressed in the cytosol, and there were effects on ligand binding. We couldn't do an association, obviously, with the development of invasive aspergillosis because we only had one patient. This one patient did develop invasive aspergillosis and unfortunately went on to die from complications of that. But we also, as I mentioned, we're looking at cytokine production, which we believe is downstream of these. So we used LPS as a control, and what we found is that our patients, I think almost all, I think it was literally one patient, their cells did not show either an IL-6 or a TNF response to LPS, whereas when we stimulated with Aspergillus, there were quite a few patients where their cells did not give a cytokine response. But you can see that in the invasive aspergillosis cohort, almost none of them did. And when we separated them out into the bone marrow transplant patients, none of them developed or produced IL-6 or TNF, whereas with the AML patients, we only had a few of those, and a couple of them did produce a bit of IL-6 and TNF. So when we did the analysis on it then, for the bone marrow transplant patients, basically if a patient's cells did not produce TNF or IL-6 in response to Aspergillus, then there was much higher chance of them going ahead and developing invasive aspergillosis. And so when we separate that all out, basically if In the high-risk patients, if a patient's cells produced IL-6 or TNF in response to LPS, but did not produce those cytokines in response to aspergillus, then 62% of them developed developed aspergillosis. Whereas with the low-risk patients, basically, if a patient came in and their cells responded and produced IL-6 or TNF in response to aspergillus, then none of them developed invasive aspergillosis. So, you know, we've gone from a 27% risk to maybe like 62% versus 0%. And then if we add on a couple of other factors, possibly like the CLR expression levels, we could get that up to 80%. So what we're actually trying to do at the moment is I'm collaborating with a clinician, and so we have to do this in Cardiff because they don't prophylactically treat the hematology patients. In Belfast, they do prophylactically treat. So I'm collaborating with a clinician at Cardiff to try and do a follow-on study with this, and we're working with a company in Cardiff as well. So this is an assay that we did on PBMCs, which is very labor-intensive, getting the PBMCs and doing this whole assay. And so we're trying to modify it and streamline it into a whole blood assay and trying to integrate other factors like SNPs that have also been associated with the risk to see if we can, you know, really get this into a set of factors that can stratify really well. And then finally, this is something we've just started on a little bit. So my colleague here, Becky Ingram, has this antigen discovery pipeline that she's been using for years to try and develop vaccines for bacteria against the escape bacteria. So we've been collaborating, trying to use this and slightly modify it to try and design or identify vaccine candidates for fungi. So what we've done is this in silico analysis part based on Candida albicans, and gotten a list of targets that should give like T-cell immune response. And then we've sort of lined that all up, against various Candida species and also against Aspergillus fumigatus. So we actually have a nice list of conserved proteins. Now, as we go down that list, if the ones at the top don't work, then we will lose some of these species, starting off with Aspergillus, and maybe Candida glabrata would be the next one we lose. But we have cloned several of these genes and expressed them, so we're now at the point where we're going to start testing these and see whether they have any effect. And then, so yeah, just to summarize then, we've shown that the CLRs can collaborate to mediate the protective effect of monocytes during Candida albicans. And as I said, we're now looking downstream, showing that the CLRs are inducing this interferon response and figuring out what some of these interferon-regulated genes are doing in response to Aspergillus and some other fungal pathogens as well. Shown that there's an early stop codon mutation in Dectin-2, and that results in unstable protein and reduced functional responses. And as I said, the one patient that we had on that did succumb to invasive aspergillosis infection. And then a potential stratification strategy, so that getting blood samples from patients, and if their cells can actually respond to Aspergillus, then the chances are they will actually do okay. If their cells do not respond to Aspergillus, then they have a much higher risk of developing invasive aspergillosis. And as I said, we're trying to move on with a follow-on study. And then, as I said, we've identified some vaccine candidates. And one slightly shameless plug here, made an exhibition for W5 here that we launched in September, kind of highlighting how bacteria develop resistance and the kind of work that we're doing in the Wellcome Wolfson for that. And it was on September, and because this is Antimicrobial Awareness Week, it's on this week until Sunday. So if you have nothing to do with your children this weekend, go and see that. And just to thank, the majority of this work was done by Aisha and James, and now Wang and Bethany are doing the follow-on studies. So thank you.